Class, this is chapter 12. We're going to cover several different topics of genetics. This will cover several labs in the lab. Let's get started. So most of the first studies on complete dominance in genetics were done by Johann Mendel. He picked a model system of garden peas. The reason he picked garden peas, they can self-fertilize. They have male parts and female parts, so they can self-fertilize. When they self-fertilize, they become true breeding. They're basically so inbred that their genetics, for their genotypes, they're all homozygous. He could also control fertilization by snipping off the male parts and painting pollen onto the female part. In this way, he could control which plants were crossed with which. So which was the father and which was the mother, essentially. He also picked pea plants because they have a ton of offspring. And this way, he could get enough numbers to have confidence in his data. And they reproduce quickly. And for all these reasons, he picked pea plants as his model system to study genetics. He did get a little lucky. Several traits like face shape or height, they are controlled by many different genes, and each gene can have different alleles. In your book, that's called continuous variation. I may refer to it as a polygenic trait, poly just meaning many, and genic meaning genes. But he got lucky with pea color because it was just controlled by one gene, and there were just two different alleles, and one was dominant and one was recessive. And he actually found several different traits within pea plants that followed this pattern, and I'll get to that in a second. So this is a portrait of Johann Mendel. He was a priest, and he was a teacher, and he was interested in science, especially inheritance. You know, he saw that children look like their parents. Sometimes they look like a blend of the parents. Sometimes they look like one parent specifically. And sometimes there's some unexpected things. Some children don't look like they belong to a family, even though they do. So he was interested in the mechanisms behind inheritance. Today he's known as the father of genetics. He did some of the first experiments. They were very organized and brilliantly done, and he kind of got lucky. So he actually had data that showed some specific laws it supported hypotheses that turned into laws later, and we'll go over those laws later in the chapter. But yeah, let's start talking about his experiments. So for all of Mendel's experiments, he took true breeding plants. That means he sort of quarantined, say plants with purple flowers, which in the book is called violet. I'm going to call it purple. So he took these plants that were creating purple flowers, and during fertilization, when pollination would occur, he would quarantine these plants. He would cover them with plastic, basically. And so they would only self-fertilize. So the purple plants would only fertilize with other purple plants or themselves. And he did the same thing to the white flowers. And he didn't know exactly what he was doing, but basically he was inbreeding them to the point where the purple plants were homozygous and the white plants were homozygous. So he then snipped all the male parts out of the purple flowers and painted pollen from the white flowers onto the female part of the purple flowers. So this way he bred the purple flowering plants with the white flowering plants. So these plants that he's breeding, those are called the P or parental generation. And he's crossing these true breeding plants, the inbred homozygotes. Those offspring are called the F1 generation, or the filial one. In his experiment with flower color, the F1 generation all made purple flowering plants. So the offspring between crossing a purple flowering plant and a white flowering plant that were true breeding, homozygous, they created a F1 generation that was 100% purple flowering plants. So he took two plants from, well, equal numbers of plants from the F1 generation, and he bred those together. So once again, he cut off the male parts to half of them, but left the female part, and then he would paint the pollen from the other half onto the female part of the half that was missing the male parts. So this way he was breeding half of them with the other, and he was controlling that breeding. And the offspring of those two F1 plants created the F2 generation offspring. And what he saw was in the F2 generation was that roughly 3/4 of these plants were purple flowering plants, and about 1/4 of these plants were white flowering plants. So that was his data. And then he wanted to try to explain his data. If you look on page 325 in the textbook, it can show you several different traits he looked at and characteristics. A characteristic is like flower color. It's the characteristic that you're looking at. Other examples would be flower position, plant height, seed texture, seed color. And then the trait would be the variations in that characteristic. So for flower color, he saw purple flowers or white flowers. For flower position, the terminal trait is where the flower is at the very end of a stem, and the axial trait is where it's not at the end, basically. For plant height's the characteristic. The two traits he saw were tall or short. He called it dwarf. For the characteristic of seed texture, He saw round or wrinkled traits. For seed color, that characteristic, he saw yellow and green seeds. So those are the two traits. So your traits can also, he called them traits, we call them phenotypes. But the characteristic, you should always call those characters. Things like hair texture, flower color, plant height, seed texture, all those should consistently be called characteristics. Traits, you can substitute the term phenotype. Anyway, if you look at that chart there, you can see how many F1 offspring he had and what the phenotype result was. And it also shows you the F2 offspring phenotypes and their sort of the total numbers he got with the offspring. And then in F2 ratio, he can show you the ratio of, for instance, violet to white, axial to terminal, tall to dwarf. And you can see those ratios are pretty close to 3 to 1. For flower color, you got a 3.15 to 1, very close to 3 to 1. For plant height, he got a 2.84 to 1. That's very close to 3 to 1. So all these ratios are pretty close to 3 to 1. And so I'm skipping a section on probability basics. The book can teach you the product rule and the sum rule. I went over that in lab, so I'm not going to cover it here. But if you have any questions about it, just email me. I'm going to skip ahead to where we start sort of explaining the data that Mendel got, which is section 12.2. It starts on 328. So before we get into this figure here, I want to go through some vocabulary. I'm going to start with gene, even though it's not really in the section. A gene is a section or segment of DNA that encodes for a protein. So it has the instructions on how to build a protein or a polypeptide. An allele is a version of a gene. So even though the gene can encode for the same protein, if it's mutated a little bit, you can get a different allele of that gene, a different version of that gene. So there would be two alleles for that gene if there's just one mutation. And so you're creating two slightly different, two slightly modified proteins, and they may behave differently. Alleles can give you different traits, like purple flowers versus white flowers, or tall versus dwarf plants, or yellow versus green seeds. So in this aspect, different alleles can give you different phenotypes depending on what type of dominance you have. In diseases, one allele could give you the disease while the other may protect you from the disease, depending on the genetics there. So let's start talking about phenotype versus genotype. So a genotype is sort of a representation of the alleles that an organism has in its DNA. Because we have homologous chromosomes, we have 2 of chromosome 1, 2 of chromosome 4, 2 of chromosome 5. We inherit 1 chromosome 5 from our mother and 1 chromosome 5 from our father. Each chromosome is going to have a copy of a gene. So say for flower color, the gene for flower color is on chromosome 5. I'm not saying that's actually true, I'm just using it as an example. So you have 2 copies for the gene for flower color. You have 2 copies of the gene for plant height. You have 2 copies of the gene for seed color. That's sort of the nature that comes from having homologous chromosomes, 2 copies of each chromosome. Now, the chromosomes may have slightly different sequences, which means that you may have the same allele on each chromosome, or you may have different alleles on each chromosome. The combination of these alleles we write as a symbol, and that is called the genotype. Geno means gene, and type means picture, so this is literally a gene picture. It's sort of a symbolic representation of your DNA. The phenotype is sort of the outward result of your genotype. So pheno means appearance and type means picture. So this word literally means appearance, picture. And this is an actual description of the outward appearance. So words like tall versus dwarf, purple versus white, flowers, yellow versus green, seeds, round versus wrinkled seeds. So those are phenotypes. Your genotypes will typically have 2 letters per gene. Later we'll get into dihybrid problems where we're tracking more than 1 gene, but for now it should be 2 letters. Whereas the phenotype should be descriptive. Either you have the disease or not, you have yellow seeds or green seeds, things like that. Now, if on chromosome 5, I said that was flower color, so on your parental chromosome 5 and your maternal chromosome 5, if you have the same allele on both, whether it's dominant or recessive, that is known as homozygous. So a zygote is the single cell result of the fertilization between a sperm and an egg. So if the zygote is homozygous, that means each chromosome 5 has the same allele for the flower color gene. Heterozygous, hetero means different, homo means same, sorry. Heterozygous, so if it's heterozygous, that means it has different alleles on the parental chromosome and the maternal chromosome 5. So you have 1 allele for purple and 1 allele for white. So that's all the terms so far. Let's do a couple of others. So when you cross 2 true breeding plants, like Mendel did, he's about to explain the data here in this figure. But whenever you cross 2 true breeding plants, that is known as a monohybrid cross. So in our parental generation, instead of looking at flower color, we're looking at seed color here. So he crossed true breeding plants that they all produced yellow seeds. no matter how you cross them together. And he had some true breeding plants that created green seeds. That means anytime he crossed them with each other, they always made green seeds. He took plants that were yellow, that were creating yellow seeds, and he crossed them with plants that were creating green seeds. True breeding plants for both of them. So that first cross he did was the parental generation, and that is a monohybrid cross because he's crossing two true breeding plants. And because of his understanding of probability and his understanding of math, he came up with this idea of a Punnett square. And well, he didn't invent it, but he used a Punnett square. And the Punnett square will use rules of probability to tell you all the possible offspring from a cross. Now, because of Mendel's understanding of genetics, based on the ratio he got a 3-to-1 phenotypic ratio for many, many traits. All the traits on page, where was it? Page 325. So you got 3-to-1 ratios on all these traits. Because of his knowledge of probability, he figured there were 2 copies of each gene. And he figured out that one was dominant over another. So in his parental generation, he crossed yellow true breeding plants right here. So he knew they were homozygotes, probably. And he crossed those with green true breeding plants. So he knew they were homozygous, probably. The yellow true breeding plants, they're only going to give you one possible gamete. And your green true breeding plants would only give you that possible gamete. And this would explain why you got a 100% ratio in the F1 generation. So if you look at that chart back on that page, 325, if you look at the F1 offspring, they're 100% the dominant trait. And this is kind of how he learned which trait or which phenotype was dominant. So here 100% were yellow. But he figured they were heterozygotes, meaning they had 2 different alleles. So they had a yellow allele and a green allele, but yellow was dominant over green. So the letter he picked was yellow, or Y for yellow being dominant. And so the recessive allele is represented by a lowercase y. And that's the allele for green. So the next thing he did was he crossed plants from the F1 generation. So he did multiple plants, but he crossed plants within the F1 generation, so offspring from the P generation. And he created the F2 generation. So these are offspring of the F1 generation. And since both parents were heterozygotes, meaning both parents had 2 different possible gametes, this was the Punnett square he got. And this would explain his 3-to-1 phenotypic ratio. He figured if there were 2 copies per gene, that this would be his genotypic ratio. So I'm going to explain dominant and recessive here, even though they've explained it somewhere else. So, like I said before, he knew the dominant trait from the F1 generation. When he crossed 2 true breeding plants, for instance, yellow seed plants with green seed plants, he always got an F1 generation where 100% were one trait, one phenotype. And that phenotype was the dominant phenotype. And the dominant allele was responsible for this. So what dominance means is that if you have at least one copy of the dominant allele, that is the phenotype you see. You see the dominant phenotype. So the dominant allele here encodes for yellow colored seeds. And the F1 offspring, all of them had at least one yellow allele. So that was the phenotype you saw. You saw 100% yellow colored seeds in the F1 generation. In the F2 generation, he sort of shows that green colored seeds is due to a recessive allele, and that you need 2 copies of the recessive allele to see the recessive phenotype. So a dominant trait is a trait where if you have at least one copy of it, that's the phenotype that you see. So if you have at least one copy of the dominant allele, that is the phenotype you see. So any heterozygotes with complete dominance, you see the dominant allele. Recessive traits, they are masked by the dominant allele. And in order to see the recessive phenotype, you need 2 copies of the recessive allele. So that is dominance and recessive. Dominant traits mask the recessive traits whenever it's complete dominance. And recessive traits are masked by the dominant trait. So in order to see the recessive trait, the only way you see the recessive trait or the recessive phenotype is if you have 2 copies of the recessive allele. So little y for seed color. What that means is that this genotype gives you yellow flowers. This genotype, the heterozygote, gives you yellow flowers. But this genotype right here gives you green, oh, sorry, I've been saying flowers, green seeds. So these 2 genotypes oh, that's a bad arrow. These 2 genotypes give you the yellow colored seed phenotype, and this genotype gives you the green colored seed phenotype. Okay, enough with this figure. So when he had plants that were making yellow seeds, he wasn't completely sure whether that plant was a homozygous or a heterozygous. So because of dominance and recessive, this genotype will give you yellow seeded plants, but also, oh, these Y's are awesome. This genotype would give you yellow seeded plants. So he invented the test cross in order to figure out what genotype he had, because this was before DNA testing and he couldn't really look at the genotypes, but he could figure out with a test cross what genotype they had. So in order to do a test cross, he crossed his yellow flowering plant with a true breeding yellow seeded plant with a true breeding green seeded plant. And he calculated basically he tried to predict the results of the cross. So if this question mark is a capital Y, this is the square you would get. So big Y, little y here, big Y, sorry, here, big Y, little y here, big Y, little y here, and big Y, little y here. So if it's homozygous dominant, you would get a 4-0, or your offspring should be 100% yellow, the dominant phenotype, the dominant trait. If this question mark is a lowercase y, let me draw my bar. This is the ratio. This is the square you get, and the ratio you get is a 1-to-1 ratio. So you get a big Y, little y here, big Y, little y here. But because this question mark is a little y, you get homozygous recessive there, and homozygous recessive there. So if you get offspring that are 100% yellow, you know your yellow plant, your yellow parent, was homozygous dominant. If your offspring ratio is 1-to-1, then you know your yellow seeded plant was a heterozygote. So this is what a test cross does. It tells you the genotype of a parent displaying the dominant phenotype. If it's displaying the dominant phenotype, you don't know if it's homozygous dominant or a heterozygote, and the test cross will tell you whether that parent showing the dominant phenotype is a homozygous dominant or a heterozygote. And the way you do that is you cross it with the recessive phenotype, in this instance green seeded plants, And then you look at the ratio of the offspring. If it's 100% dominant phenotype, then you know you had a homozygous dominant parent. If it's a 1-to-1 ratio in the offspring, then you know you had a heterozygote parent. So that's the test cross. Okay, so the next tool that we're going to go over after test cross is going to be a pedigree. And you can analyze a pedigree and tell me whether a trait is being inherited dominantly or recessively. And you can have autosomal dominant or recessive. Autosomal just means it's not sex-linked. And all your problems in this class are going to be either autosomal dominant or autosomal recessive. You can also have sex-linked dominant or sex-linked recessive. You can also have X-linked or Y-linked. But we're not going to analyze those types of pedigrees in this class. So when I ask you to analyze a pedigree and I ask you how a trait is inherited, you need to figure out whether it's autosomal dominant or autosomal recessive. And I'll tell you the way you do that. So the way you do that is you start at the bottom of the pedigree. Every time you see an affected child, you look at their parents. So here from this child, one of her parents had the trait, but if you look at this child, neither of her parents had the trait. Because you have this one instance where the parents did not have the trait, but one of the offspring did. That means that is autosomal recessive, because one child on this pedigree had parents that did not have the trait. So they were not affected. So typically in these pedigrees, some other things you need to go over. Oh, wait. So if it's dominant, every time a child has the trait, at least one of the parents has the trait. So if a pedigree shows that pattern where every time the child has the trait, at least one of the parents has the trait, then it's autosomal dominant. This image right here that we're looking at, we had at least one family where the offspring had the trait, but the parents did not. So that is autosomal recessive. That's the type of inheritance. Okay, so some other things about pedigrees. Squares represent males. Circles represent females. Unaffected are typically unshaded. I don't like this figure because they're color coding. But typically, universally, with pedigrees, unaffected people are unshaded. So it'd be an empty circle or an empty square. I know those look identical, sorry. Now, affected individuals tend to be shaded. So they're either colored in or they might, they're shaded in some way. So unaffected people are typically unshaded. They just have empty circle or square. And affected people are typically shaded. So that's the pedigree. That's what you need to be able to tell me. What's male, what's female, what's affected, what's unaffected, and then how is it inherited? Autosomal dominant or autosomal recessive? So now we're going to get into some exceptions to complete dominance and recessiveness. The first one to go over is incomplete dominance. If we talk about carnation color or snapdragon color or hair texture, you should start thinking incomplete dominance. If you cross red flowering snapdragons with white flowering snapdragons, this picture is showing the intermediate phenotype, the pink phenotype. In incomplete dominance, the heterozygote is a blend of the two homozygous. Or another way to say that is that an individual that has two different alleles will display an intermediate phenotype between the two homozygotes. So, like I said, we're going to use superscript to denote that this is a special exception to complete dominance. We're going to use a capital C for color. And then the superscript, we'd use an R for red. That's a very tiny R. R for red, and for our white colored snapdragons, we'd use a CW for white. So if the plant is red flowering, its genotype would be CRCR. If the plant is white flowering, the genotype would be CWCW. And if it's pink, like this plant here, right here, this would essentially be its genotype, CRCW. For multiple alleles, they give the example of chinchillas, chinchilla coat. But when you think about animal coat, I really want you to think about epigenetics. And this chinchilla example has lots of things going on, so it's a little confusing. So instead, for codominance and multiple alleles, I want you to look at ABO blood types. Codominance means that two different alleles are expressed at the same time on the same organism in the heterozygote. For like ABO blood types, the blood type AB has a IAIB genotype. And multiple alleles simply means that there are more than two alleles for the genotype. For ABO blood types, one allele would be IA, another allele would be IB, and the third allele in that example is a lowercase I. So that's all I'm going to go over incomplete dominance and multiple alleles and codominance in lecture. I cover it more in lab. If you have any questions over it, just email me. The next thing we need to go over are sex-linked or X-linked genes. For most of the examples you'll see on either the lab practical or the lecture exam, these are all going to be X-linked problems, genetic problems. One thing I need to talk about is the concept of hemizygous. Males in sex-linked genes are considered hemizygous because they only have one X. Males have one X and one Y, so that equals male. When you have a female, you have 2 copies of the X. And since we're looking at X-linked traits, the female can be homozygous or heterozygous. But the male is essentially hemizygous. That just means that he only has one X. So he can never really be homozygous or heterozygous because he only has one X. He is considered hemizygous. That means he only gets one copy of an allele from X-linked genes. So in this sex-linked problem, they're looking at the eye color of flies. The wild type, the trait that you see in the wild most often. So the dominant one is the red eye. So red eye is dominant. And they represent it with a big W for wild type. Now the eye color you see not very frequently in the wild is white. And so white is a mutant, and they represent that by a lowercase w. It's important to notice in this example that W stands for wild type, not white. And wild type is the trait that you see most often in the wild, not in a lab setting. So in this example, they crossed red-colored eyed males with white-colored eye females. And they saw a 1-to-1 segregation of eye color. But interestingly, all the red eye flies were female, and all the white colored eyes were male. So it switched from the parents. And so this was one of the first experiments that showed that some genes are sex-linked. If you're crossing... Okay, so let's look at the genetics. So your female was homozygous recessive. So she was X little w, X little w. Her possible gametes, you can either have an X little w or an X little w. The male was an XY, and his 1X was a capital W, and that's why he has the red eyes. So his possible gametes are X capital W or Y. If you do the cross, his X with the big W goes to the females. And they only need one big W to get red eyes. The males, however, got their X from their mom, and they get their Y from their dad. And the mom only had X little w, which gives you white eyes. So that's why both of the males ended up with white eyes. So your genotypic ratio, you have 2 of these and you have 2 of these. So it would be a 1-to-1 ratio. So it would be 1 X big W, X little w, to 1 X little w, Y. And the phenotypic ratio, you would have 1 red-eyed female to 1 white-eyed male. Now this term carrier comes up often in sex-linked genetics problems. And carrier just means that the female is a heterozygote. So she would be, in this instance, X big W, X little w. She does have red eyes because she has at least one dominant allele for eye color, for the wild type eye color. But she would also be described as a carrier. So because she has one copy of the recessive allele. So even though phenotypically she's normal, she's wild type, she has the red eyes, genotypically she's a carrier. She has at least one copy of the recessive allele. So in X-linked disorders, one allele is dominant to the other, but males are hemizygous, so they cannot be protected by the homologous chromosome nature of most genetics. Males only have one X chromosome, so they do not get 2 copies. So if they have one recessive copy, one recessive allele, that's enough to give them a disease or a disorder. And some examples of X-linked disorders include color blindness and male pattern baldness and hemophilia. A carrier mother, I just want to note again, is unaffected. She does not have the disorder, but it means she has a recessive allele. And she gives her sons their X. So if she's a carrier, her son has a 50-50 shot of being affected by this disease. As long as in these X-linked things, if the father is unaffected or normal, then both of his daughters are going to be normal. However, because the mother is a carrier, at least one of their daughters will end up being a carrier. So daughters or females are protected by X-linked diseases because they have 2 copies of an X, but sons are not very well protected against X-linked diseases because they only need 1 copy of the X. Another thing to go over is recessive lethal. And this is when the recessive allele results in lethality in an organism. So these individuals may abort in utero. They may die in childhood, or they may die possibly during birth, which is very sad. But in this pattern, a lot of times you'll see if it's X-linked, for example, you'll see daughters being born at a way higher rate than sons. And the genotypic ratio that you observe in the offspring would be a 2-to-1 ratio because you do not see the affected sons. They are killed at some point in the life cycle. The worst are dominant lethal. And all offspring that have the dominant allele will eventually inherit the disease. The example is Huntington's disease. And these people live slightly past the fertile stage of their life. But if they have at least 1 copy of this allele, they will get the disease and they will die in their 40s to 50s. So know what lethal means. Lethality just means that if they get the disease, essentially, they die at some point. Recessive lethal, they only die if they have 2 copies of the recessive gene, or if it's X-linked, the son that inherits the recessive allele would pass away, and that will give you a 2-to-1 ratio in offspring. A dominant lethal, these genes or these diseases will kill you if you get a single copy of the dominant allele. So you could be homozygous or you could be heterozygous, but it still lead to lethality. So the next topic we need to go over is a dihybrid cross. And then I'm going to skip forward to epistasis. And I will be pretty much done with this chapter. And I'll tell you what you can skip in this chapter. So you do need to know Mendel's laws that he got from doing all this data. The law of dominance really is talking about complete dominance. And it says that in a heterozygote, 1 trait or 1 allele, excuse me, one trait or 1 allele will essentially conceal or mask the other trait or allele. The other law he came up with was the law of segregation. And this says that there are 2 copies of each gene, and that whenever you form gametes, whenever you form sperm or egg, these copies separate. And so this is why in the past, when we've looked at a gene, the genotype has been symbolized by 2 letters. But when we look at the possible gametes, you're looking at only 1 letter. So that's the law of segregation. It's just basically saying that we have 2 copies of each gene, and that whenever you form gametes, they're separated. His third law was the law of independent assortment. And this pretty much means that genes are unlinked whenever they're sorting. And his dihybrid experiments sort of explain that. So let's get into his dihybrid experiment. And then I'll get back to the law of independent assortment. So in his dihybrid experiments, he started to track 2 characters. So in these plants, he was looking at seed color, but also seed texture. And his true breeding plants had the dominant phenotype. They are homozygous dominant for both seed color, yellow over green, and seed texture, round over wrinkled. And he crossed those with the recessive true breeding plants. So these plants had green seeds that were also wrinkled. So as P generation, you had plants with yellow round seeds, and you cross those with plants with green and wrinkled seeds. His F1 generation, all were yellow, all created yellow and round seeds, but he now had a dihybrid. So the di means we're tracking 2 genes, and the hybrid part means heterozygote. It came from crossing true breeding plants, and the true breeding plants are homozygous for their traits. So this F1 generation is pretty much as predicted. 100% of them had yellow seeds that were round. So then he crossed plants from the F1 generation, and he got the F2 generation. Now, depending on which ratio he got here, the genes would either be linked or unlinked. Linked meaning they were inherited together, and unlinked meaning they were inherited separately, or they had independent assortment. So when you're doing a dihybrid problem, both your parents look like this. So you have a big Y, a little y, a big R. Even though R's look different enough, I'm still gonna do the bar for it. Okay, so the hard part about 2 gene problems is making the gametes. You can't just split them anymore. You have to come up with all the possible gametes. So the way I do that is first I take the first copy of the first gene, and I mix it with the first copy of the second gene. So the red one is a big Y, a big R. Second, I take the first copy of the first gene, and mix it with the second copy of the second gene. So the blue one is a big Y, little r. Then I take the second copy of the first gene and mix it with the first copy of the second gene. That gives you a little y with a big R. And with monohybrid problems or one gene problems, you always want to put the capital letter first. But with two gene problems, I think it's more important to keep the genes in order. So yellow before, or seed color before seed texture. So that's why I put the little y first. Lastly, I'm going to take the second copy of the first gene and mix it with the second copy of the second gene. And that would give us a little y with a little r. So for this individual, these are all the possible gametes. And this means you need a 4x4 Punnett square. And you'll notice I would do this the exact same way, but the book has sort of laid down the alleles in mirror image. So you have big Y, big R first. I'd do this slightly different, but that's okay. And then they did little y, big R second on both. And then they did big Y, little r third on both. And little y, little r fourth on both. Actually, I don't like how they've done this. I will show you on lab a little bit easier way to do this. Anyway, so your phenotypic ratio, these are yellow and round. These are yellow and round. These are yellow and round, and those are yellow and round. These are yellow and round. These are yellow and round. These are yellow and round. Those are yellow and round. And those are yellow and round. So that's 1, 2, 3, 4, 5, 6, 7, 8, 9 that are yellow and round. So 9 yellow and round. And then this is yellow and wrinkled. That's yellow and wrinkled. That's yellow and wrinkled. So you have 3 yellow and wrinkled. This is green but round. This is green but round. And this is green but round. So you get 3 green and round. And then, what color have I not done yet? These right here. You only have 1 that are green and wrinkled. So that's your 1 that's green and wrinkled. So when you're doing a two gene cross with dihybrids, so they're heterozygotes for both genes, this is the phenotypic ratio you'll get: 9, 3, 3, 1. And because you got this ratio and not your typical 3-to-1 ratio, that means these genes are unlinked and they assort independently. So independent assortment is just saying that genes for different characters are inherited independently. They are not inherited together. And the way he did that was with these dihybrid experiments. Don't worry too much about this section. That's talking about probability method or forked line method. I wouldn't even really worry about the rules for multi-hybrid fertilization. It's important to know that some genes do violate. Hold on. Let's go to the next slide real quick. So some genes do violate independent assortment. Some genes are linked, and it's based on how close they are on the same chromosome. So for genes to be linked, they have to be on the same chromosome, first off, and then they have to be pretty close together. Their loci, their locuses have to be pretty close together on the same chromosome. And the most common way to violate independent assortment is with crossover. And I'll go over that a little bit more in the next chapter, so that's all I'm really going to say about it here. If genes do not segregate or assort independently, meaning they are linked, then it's usually because of crossover. Don't worry about this. Okay, so this is the last figure I'm going to go over. And it's explaining the concept of epistasis. Epistasis means that one gene affects the expression of another gene. And if we're talking about Labrador coat color or mouse coat color, we're typically talking about epistasis. We'll go over some epistasis problems in lab. But sort of the definition of it is that a gene at one location, one locus, affects a gene at a different locus, whether it's expressed or not. So for example, we're looking at a two-gene problem, or a dihybrid cross. One gene we're tracking is the C gene, and it is responsible for pigment production. If it has at least one capital C, that means you're going to get color. But if it is homozygous recessive for the C gene, that means there's no color. With coat color and skin color, that is known as albino. The other gene we are tracking is the A gene. It's referring to the dominant coat color, which is agouti. Agouti is pretty much tan. The recessive, so a big A, if it has at least one big A, you're going to get the agouti color. If there's a little a, then you're going to get a black color code. It's really super dark gray, but we'll call it black. So if you do the dihybrid cross, crossing this mice with this mice, these are the possible gametes you get for both of them. Big A, big C, little a, big C, big A, little c, little a, little c. Those are the possible gametes you can get. So after you play bingo, the first thing you want to look at is the C's. So what we're looking for is a homozygous recessive C, and those will tell you the albino mice. So we have two lowercase c's here, 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 and here. Those four possibilities have white coat color. The rest of these have at least one big C, so then you look at the A. If it has at least one capital A, so if it has at least one capital A, the mouse is going to be agouti, or this brown color. So we have two A's there, or two capital A's there, one there, two there, one there. Actually, let me use another color, sorry. It's there, 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 there. There's a capital A. Here's a capital A. Here's a capital A. Capital A. Capital A. So all those mice, or 9 out of the 16, are going to be agouti colored, or this brown color. Lastly, let's see, let's do blue. If they are homozygous lowercase a, then you'll get this black color. So that guy's two little a's, and two little a's. So 3 of the 16 possibilities are black, and from before, 4 out of 16 are albino. Okay, so that is epistasis. Like I said, I'll go over a couple more examples in lab. So you should have a pretty good idea of epistasis, but once again, just to define it, it means that a gene at one locus affects the expression of a gene at another locus. The last point I want to make, I can explain this a lot better by doing practice problems in front of you than I can just lecturing about these figures that are already made. So if you have any questions, email me, and you'll get a lot more out of this when I do practice problems in front of you. But I still went over quite a bit of the vocabulary, and I felt like I defined those pretty well. So make sure you know your vocabulary from this lecture, and then know how to do each type of problem. So monohybrid cross, incomplete dominance, know a test cross and a pedigree, know incomplete dominance, codominance, multiple alleles, know examples, know what hemizygous means, know what an X-linked disorder is, be able to do an X-linked problem. Know about Mendel's laws, know how to do a dihybrid cross, and know how to do an epistasis problem, and know what epistasis is. So that's pretty much it for this chapter. It was actually a lot, sorry. So yeah, this chapter does cover a lot. But chapter 13, I'm going to condense quite a bit and just highlight exactly what you need to know. So even though 12 is huge, 13 is quite tiny. So we'll do that next.